So, good morning to all of you, students of BT First Sem. Today, I am going to discuss with you about an important topic which is given on your biotechnology and human welfare. That is your GE paper, General Elective paper. And the name of the topic today that I am going to discuss with you is about the nitrogen fixation. Now, only nitrogen fixation has been written on your syllabus. There is no so much there is not much information given about the or the details has not been given, so for that I have to cover up little bit overview the whole topic related with the nitrogen fixation. And as it is given on biotechnology and human welfare, so I have to say some aspect of nitrogen fixation related with biotechnology. Okay, so let's get started now. So before going into this, we have to define this term of nitrogen, biological nitrogen fixation, right? So normally, what is biological nitrogen fixation? If I say, it is saying that nitrogen is an important essential element of many biomolecules, right? Okay, as you know, nitrogens are very much important biomolecules which are needed for your nucleic acid production, for amino acid production, right? Nucleic acid like DNA or RNA. If you see the structure of DNA and RNA, the nitrogenous bases, the purines and the pyrimidines, okay, which forms the main nitrogenous base of DNA and RNA, they are made up of mostly of nitrogens, right? So nitrogen is required for us for the synthesizing of nucleic acids and also amino acids, which are the building blocks of proteins. Although nitrogen is the most abundant gas, about 80% in the atmosphere, neither animals nor plants can use this nitrogen to synthesize their biological compounds. So, as nitrogen is also available in the atmosphere. About 80% of our atmosphere is having nitrogen, but still either the animals or plants cannot utilize that atmospheric nitrogen, cannot use that nitrogen to synthesize their nucleic acids, to synthesize their amino acids, which are the biological compounds. Okay. However, there are certain microbes, or you can say microorganisms, on which the living plants and animals are dependent on that microorganisms to bring that atmospheric nitrogen into their biological system. So there are some certain microbes which can allow that atmospheric nitrogen to be introduced into the biological system of living organisms. Like in your junior classes, you have studied about rhizobium. Rhizobium. Sorry for that. Okay, I have to change the color now for you to understand easy. So rhizobium, Rhizobium. So rhizobium species, these species can associate with the roots of leguminous plants like pea, like your soybean, like your gram, okay, like your um, tamarind. Okay, these are some leguminous plants because the roots of these plants they form legumes like this. If I show you the roots of these plants, if this is the roots of the plants, they form legume like structure like this, nodules. You can say legumes or you can say them as nodules like structure. Now, within these nodules, within these nodules, rhizobium associate with the leguminous plant on the roots. Okay, so rhizobium associate with these roots of leguminous plants, and they have the ability that they have some genes, they have some enzymes or gene encoded enzymes, better to say, which will fix that atmospheric nitrogen, in the soil, and will provide that nitrogen to the plants. In exchange, rhizobium will get the nutrients from the plants. This type of association is called symbiotic nitrogen fixation or symbiosis. Well, some microbes are also there which present independently in soil. There are some microbes which present independently in soil. That means they don't depend on plants or animals, and they can fix the atmospheric nitrogen in the soil. Well, the nitrogen will not be found directly. It will be found in the form of compounds, like you can find them in the form of nitrite or in the form of nitrate. You will find out, or in the form of ammonium ion, right? So there are some microbes which can be used, which are actually which can use this nitrogen to synthesize their biological compounds. However, there are certain microbes on which the living plants and animals are dependent to bring that nitrogen into their biological system. So, with the help of microbes, that means via microbes, we are allowing that atmospheric nitrogen to be fixed into the biological systems of plants and animals, so that the plants and animals can use that nitrogen for their formation of their biological compounds like nucleic acids, amino acids, okay, which are the building blocks of proteins. Okay, so I hope you have understood this part, what I have tried to say. Now, the phenomenon of fixation of atmospheric nitrogen by microorganisms is known as diazotrophy. So, the process by which atmospheric nitrogen is fixed, okay, into the soil or into the system of the organism is known as diazotrophy. Okay, and those organisms who are fixing that atmospheric nitrogen are known as diazotrophs. Okay, so these are known as diazotrophs. So those microorganisms which are fixing that atmospheric nitrogen in the soil to provide that nitrogen um, by the living organisms, so that they can synthesize their biological compounds, are known, this process is known as diazotrophy. But the microbes which are involved in this diazotrophy process are known as diazotrophs. Diazotrophs are biological nitrogen fixers and are mostly prokaryotic in nature. So most of these diazotrophs, that means those who fix the atmospheric nitrogen in the soil, are known as diazotrophs, and these diazotrophs are prokaryotic in nature, means bacterial in nature. They are mostly unicellular. Okay, so this atmospheric nitrogen fixation, or as we say biological nitrogen fixation, is strongly related with your nitrogen cycle as given in this bottom page. So we have to discuss about the nitrogen cycle a little bit in order to understand the way of nitrogen fixation by the diazotrophs. Okay, now there are also the diazotrophs are of two types: free living and symbiotically living. That means some microbes they associate with the living systems, they associate with the plants and animals. Okay, whereas some diazotrophs they live freely in the soil and they freely carry out the nitrogen fixation. So let us first discuss about the nitrogen cycle. Now, there is an outline given on nitrogen cycle on the next page. So I'm moving to the next page, okay, for you to understand about this nitrogen cycle. Well, see, this is the nitrogen cycle. This is an outline of nitrogen cycle. Normally, your nitrogen is entering in the soil. The nitrogen is entering in the soil with deposits of dead plants and animals. Okay, so when the animals die or plants get died, the nitrogen enters to the soil. Okay, from that dead animals and plant waste. They have repeatedly written animals and animal wastes like urea, okay, in the form of urine also, right, because it contains urea, which is the source of nitrogen. Now these dead animals and animal wastes, including the plants also, these waste materials like the proteins and the urea are decomposed by various soil bacteria to form ammonia and other products. Listen very carefully what I am trying to say. So the dead animals and plants, okay, they or the waste matter of the animals are decomposed by this bacteria. They are decomposing by some bacteria, by some soil bacteria, so that they can convert them into ammonia. They can convert into ammonia and other products as well, right? Now this ammonia or NH3 is then converted into nitrite. This ammonia NH3 is then converted into nitrite, and then into nitrate. Okay, so they have little bit mistakenly they have gone through this. They are decomposed and they form nitrite first and then into nitrate. Okay, nitrite is NO2, okay, nitrite, and then into nitrate NO3. So they have written right, but the spelling is little bit mistake, okay. And then now this is also done by certain other species of bacteria like nitrosomonas species, nitrobacter species, okay. So nitrosomonas, nitrobacter, they carry out the conversion of ammonia into nitrite and nitrate, okay. Okay, now this nitrate is further degraded by various microorganisms to release the nitrogen in the atmosphere. So, okay, so this nitrate is then degraded by other microorganisms which will eventually release that nitrogen that enters the atmosphere. Okay, so this is the another way how nitrogen is getting recycled. So first, what we have said, animals, the waste of animals, then various dead plants, they or the animal waste in the form of urea, in the form of urine, okay, or fecal matter, they that contain organic matter, okay, at the same time it also contain nitrogen source. Okay, now these waste materials are further decomposed by bacteria to form ammonia. Okay, now this ammonia is further degraded or converted into nitrite and then into nitrate. Now this conversion, that is conversion of ammonia into nitrite and then into nitrate, it is done by certain microorganisms like nitrosomonas, nitrobacter. Okay, Now this nitrate is further degraded by various microorganisms so that it can release that nitrogen in the atmosphere. Okay, it can release the nitrogen in the atmosphere. Now the same atmospheric nitrogen is actually used up by nitrogen fixing bacteria. So now how you will fix the nitrogen? The nitrogen which release into the atmosphere, this atmospheric nitrogen is now fixed. This atmospheric nitrogen is now taken into the soil again by nitrogen fixing bacteria, now which are known as diazotrophs. They are known as diazotrophs. Now nitrogen fixing bacteria are of two types. One types of bacteria, they symbiotically associate with the living organisms in order to fix that nitrogen. Okay, known as symbiotic nitrogen fixers. And another type of microorganisms are there which freely they live in the soil and directly they fix the atmospheric nitrogen. So this atmospheric nitrogen taken up by this nitrogen fixing bacteria, okay, which are found on the roots of leguminous plants, and they they allow this nitrogen to be converted into ammonia. Okay, could be converted into ammonia and various other types of compound, so that we it can easily be used by the plants and used for the synthesis of biomolecules like amino acids, like nucleic acids, which is the DNA and RNA. Now, as the animal consume that leguminous plant as food, the animals are using this leguminous plant as food. Plants are synthesizing the nucleic acids. Plants are synthesizing the biological molecules from that from that fixed nitrogen. Okay, which was actually fixed by the bacteria. Now these plants were eaten up by the animals, so the animals are also consuming that, also utilizing that nitrogenous compound found in the plants, which were previously fixed by the fixed by the microbes in the soil. Okay, now as the animal consume this leguminous plant as food, the nitrogen cycle is completed, because again the animal will produce waste, again the animal will die, the plants will also die. It will be decomposed by various bacteria, and the nitrogen found in the animals will be utilized by the bacteria to produce ammonia. This ammonia will be further converted into nitrite and then nitrate. Okay, they will be further converted into nitrite and then into nitrate, which are then further this nitrate will further, what will do, they will be degraded by various microorganisms again, and they release that nitrogen in the atmosphere. Again that nitrogen will be utilized, atmospheric nitrogen will be utilized by the diazotrophs, which are the symbiotic and asymbiotic nitrogen fixers, which will further utilize that atmospheric nitrogen and converted that atmospheric nitrogen into useful nitrogen compounds, which can be used up by the plants, which associate with the microbes, symbiotically associate with the microbes, or the free living nitrogen fixing bacteria. They directly fix that atmospheric nitrogen in the soil, and within the soil the plants are utilizing that nitrogen source. Okay, these plants now making that using that nitrogen for the formation of their biomolecules like nucleic acids, amino acids. These plants are now eaten up by the animals as a result of food chain. As a result of food, the animals are getting that nitrogen indirectly from the plants via microbes. Okay, and in this way your nitrogen cycle is wholly completed. But we are now keen on interest on this what, nitrogen fixation. We are not interested in this part, because this part is how the nitrogen is going back to the atmosphere has been mentioned. We will be looking into this part, that how the microbes are utilizing, they are using that atmospheric nitrogen, okay, to be fixed into the soil, okay, and used by the plants, which are further consumed by the animals to get that same nitrogen. Okay, so now we will discuss about the nitrogen fixing bacteria. So it is estimated that 50% of the nitrogen needed by the plants come from nitrogen fixing bacteria. Okay, so most of the nitrogen needed by the plants comes from the bacteria who fix that atmospheric nitrogen into their soil. Now there are two types of nitrogen fixing microorganisms. One type of microorganisms are called symbiotic and another type of microorganisms are called asymbiotic. Normally symbiotic symbiotic nitrogen fixers symbiotic nitrogen fixers are those which associate with the roots, which associate with the living organisms, living things like the rhizobium. They associate with the roots of leguminous plants, right? And they carry out the nitrogen fixation. In exchange, the plant will provide that symbiotic nitrogen fixing bacteria the nutrients for their growth and nourishment. The asymbiotic nitrogen fixers are those microorganisms which fix nitrogen, okay, from atmosphere into the soil, but that microbes do not depend on the nourishment from the plants. Okay, they live independently in the soil and they carry out the nitrogen fixation in the soil. Okay. Okay. Again, I am repeating, this part is nitrite and this part is nitrate. Don't confuse. They have written some mistakes in these words, right? This is a printing mistake. Now let us see about the two types of microorganisms which uh, carry out the nitrogen fixation. One is called asymbiotic nitrogen fixing microorganisms. So what they are trying to say about asymbiotic nitrogen fixing microbes? Let us see. So let us discuss about this asymbiotic nitrogen fixing microorganisms. It is saying that the gaseous nitrogen of the atmosphere is directly and independently utilized to produce nitrogen rich compounds. So, the nitrogen which is found in the atmosphere, they can be directly used by microbes independently without dependent on other plants or other living organism. They directly fix that atmospheric nitrogen from the atmosphere to the soil. When these non-symbiotic organisms die, that means the microbes which are living freely in soil, those microbes, if they are utilizing, they are fixing the atmospheric nitrogen from atmosphere to the soil. Now microbes have a short lifespan, they will die eventually. So when they will die, they enrich the soil with nitrogenous compound because they are fixing the nitrogen in the soil, obviously for their beneficial. Okay, but when for making their own nucleic acids, amino acids, etc. But when that microbes die, that nitrogen compounds remain in the soil and the soil is enriched with that nitrogenous compounds. Now there are different types of species which are found in the soil that freely fix the nitrogen from atmosphere to the soil. They do not symbiotically associate with the plants. They remain independently in the soil. Several species of bacteria and fungi can do this job. Let us see example of some asymbiotic nitrogen fixing organisms. Clostridium, Clostridium pasteurianum. Clostridium pasteurianum is a such type of microorganism where Clostridium genera are some certain dangerous, like Clostridium tetani, which cause tetanus. Okay, that's why when we have a cut or wound in the field, we quickly, or if it's a severe cut, then we quickly go to get that anti-tetanus injections, right? Okay, but not all Clostridium are dangerous. Like Clostridium pasteurianum, it is a very beneficial microorganism because it freely, without the help of any living systems, they live freely in soil and they fix the atmospheric nitrogen. And when these organisms die, they enrich the soil with nitrogen compounds. Then Azotobacter crooccum. Azotobacter crooccum is also a beneficial asymbiotic nitrogen fixing microbe. The mechanism of nitrogen fixation, the mechanism of nitrogen fixation by asymbiotic bacteria is not clearly understood. It is believed that nitrogen is first converted into hydroxylamine or ammonium nitrate and then incorporated into the biomolecules. Now, the process of asymbiotic nitrogen fixation, which is done by free living bacteria, has not been clearly understood that how the microbes are fixing the atmospheric nitrogen in the soil. Okay, we have just studied, researches has been done about the symbiotic nitrogen fixers. That means those microbes which symbiotically live with the plants, or you can say living organisms, and they fix the atmospheric nitrogen. Their mechanism has been understood clearly, but the asymbiotic nitrogen fixing microbes, that how they directly fix the atmospheric nitrogen in the soil, has not been clearly understood. It has only been said that it has only been said that this nitrogen is first converted into hydroxylamine. The nitrogen found in the atmosphere is first converted into hydroxylamine or ammonium nitrate, and then they are incorporated to the biomolecules like your proteins, nucleic acids, etc. Let us see the symbiotic nitrogen fixing microorganisms. Okay, now these microorganisms live together with the plants. Okay, so normally symbiotic nitrogen fixation or the symbiotic nitrogen fixers, or you can say symbiotic nitrogen diazotrophs, they live with together with the plants in a mutually beneficial relationship in a phenomenon known as symbiosis. So, symbiosis is a term where you can see the one living organism is dependent on other living organism and they are mutually helping each other. Here, how they are mutually helping, the plants are providing nourishment to the microbes, the symbiotic nitrogen fixtures, they are providing nourishment, and in exchange the microbes are fixing that atmospheric nitrogen in the soil and providing that nitrogen to the plants. So it is a type of mutual beneficial relationship. Okay, there are also some other types of relationship that occurs between the microbes, like parasitism, where a parasite lives in the lives in our gut, and they are using the nourishment from us, but they are not benefiting us. That is the host parasite relationship. There is also other relationship like your prey predator relationship, where prey is the animal which will be used as food by the predator. So that type of relationship is commonly found among the lions, among the tigers, right? So in the same way, here it is a symbiotic relationship where mutually one living organism is mutually helping the other organism and vice versa. That is called symbiosis. The most important microorganism involved in this symbiosis belong to two related genera, namely Rhizobium and Bradyrhizobium. So the two important microbes which are commonly involved in symbiosis are, some genera has been given, Rhizobium and Bradyrhizobium. Now these are the genera names. Species name totally has not been mentioned. So Rhizobium species. So the species related with Rhizobium genera, or the species related with Bradyrhizobium genera, they symbiotically associate with the plants and carry out the nitrogen fixation, and in exchange they get the nourishment, the nutrients from the plants. These symbiotic bacteria, also referred to as nodule bacteria, okay, they are also called nodule bacteria because they form nodules in the roots of leguminous plants. They form legume like structure that I have shown just little bit ago, just like this. If this is the roots, if these are the roots, they form legume like this. They form legume like this. Nodules, right? These are called nodules. These are done by the bacteria. Special genes are there known as nod genes, NOD. Okay. Then there are also some nitrogen fixing genes as well, known as NIF genes. So let us see more about this part. So Rhizobium and Bradyrhizobium are two genera which carry out this symbiotic nitrogen fixation. These symbiotic bacteria, also referred to as nodule bacteria, and they are gram negative. They are gram negative, depend on the cell wall arrangement. We name them as gram positive and gram negative bacteria. So they have a different type of cell wall arrangement called gram negative bacteria. Flagellated, that means they have a flagellar tail which helps them to move, okay, from one place to another place, and are rod shape, just like bacillus species. The host plants harboring these bacteria are known as legumes. Now, those plants which are allowing this Rhizobium, Bradyrhizobium, that is symbiotic bacteria, to associate with them, those plants which are allowing this bacteria to live with them are known as legumes or leguminous plants. Example of such leguminous plants are your soybean, peas, beans, alfalfa, peanuts, and clover. Well, tamarind is also has found that have they have that nodule formation in their roots. It has been observed nowadays. Okay, so these plants are called legumes plants because they form nodule-like structure, known as legumes in their roots, and on that legumes, they allow this bacteria to live. Okay, and they carry out the nitrogen fixation. Okay, next, each one of these species of Rhizobium and Bradyrhizobium are specific for a limited number of plants. Now, these Rhizobium or Bradyrhizobium, if we see in case of species organization, so they associate with limited number of plants. If you see, okay, which survive as natural hosts. If you see in this table, you can see here very clearly, that different species of rhizobium are found, like Rhizobium leguminosarum, saram. It will be soram, like Rhizobium leguminosarum, I think it will be. Little bit printing mistake has been given. So Rhizobium leguminosarum, this bacteria is found on the roots of pea broad bean. So it is species specific. It is limited. This species of Rhizobium, Bradyrhizobium are specific for limited number of plants. Then you see Rhizobium meliloti. These species of Rhizobium associate with the alfalfa plant only, not on other plants. Then Rhizobium fredi. These species of Rhizobium associate with the soybean. Rhizobium LT. This Rhizobium is associated with the mung bean, kidney bean, etc. Then comes the Bradyrhizobium japonicum. They strongly associate with the soybean plant, not on other plants. So they are species specific, right? And they survive, and the species they are surviving as natural host. It is now clearly known that these bacteria do not interact with plants other than the natural host. So if you try to suppose if you try to associate this Rhizobium leguminosarum with uh, lotus, suppose it will not associate. That means they are host specific. Okay, so these bacteria do not interact with the other plants other than the natural host. So if they are specific for particular host plant. Okay, now this symbiotic relationship, the relationship between the symbiotic bacteria and the legumes, that is a leguminous plant, is well recognized nowadays. Scientists have done various experiments based on this, and they have found a good way that how they associate with the plants, how they fix the nitrogen, and how that nitrogen is utilized by the plants. Okay, it has been said that on the roots of legumes there are certain number of nodules which are swelling like structure. Again, I repeat, it is saying that it is saying that on the roots of legumes there are certain number of nodules which are swelling like structure, round swelling like structure found on the roots, in which the Rhizobium species live, or you can say thrive. So within these nodules your Rhizobium species survive. Also the other symbiotic nitrogen fixers. These bacteria trap the atmospheric nitrogen and synthesize the nitrogen rich compounds. So these bacteria, what they do, they trap the atmospheric nitrogen from the soil and synthesize the nitrogen rich compounds like amino acids, proteins, which will be used up by the leguminous plants. At the same time, these legumes will supply important nitrogen compound for the metabolism of rhizobia. So very wellly it has been mentioned that on the roots of the leguminous plant there are small swelling like structure called nodules, where in within that nodules the rhizobium survive, and also the other symbiotic nitrogen fixers. These bacteria trap that atmospheric nitrogen and synthesize the nitrogen rich compounds to be used up by the leguminous plant. At the same time, legumes or the leguminous plant supply important nitrogen compound for the metabolism of rhizobia. And it has been mentioned that growth of the leguminous plant, if we grow the leguminous plant, it has been known to enrich the soil fertility. Leguminous plant generally enrich your soil fertility This is due to the fact that the concentration of nitrogen compound in the soil increase as a result of presence of these symbiotic bacteria. So more, if we plant leguminous plant, that means more symbiotic bacteria will be heavily populated on that leguminous plants, and thus it will enrich the soil with nitrogen. This makes the soil fertile, because we know that plant needs all of the nutrients, but mainly three nutrients are essential for the plants: nitrogen, phosphorus, and potassium. NPK. So we are providing nitrogen to the plants biologically, naturally, without using any chemical fertilizers. So it will enrich the soil fertility if we plant more and more leguminous plants. Okay. Now this is for this reason that normally nitrogen fertilizers are not needed in the fields of cultivated legumes. That means those farmlands, those agricultural lands where we are culturing, where we are growing the leguminous plants, they are in that region. We don't need any artificial chemical nitrogen fertilizers, because they are already biologically fixing the nitrogen in the soil, because within their legumes there are symbiotic nitrogen fixing bacteria which fix the nitrogen in the soil. Okay. Well, asymbiotic nitrogen fixers can also be cultivated and distributed to the lands through the help of microbiological techniques and biotechnological approach also, because we want to improve the strain, we want to improve the strain of that bacteria by by modifying their metabolic system, by introducing some genes which will produce more nitrogen fixing capability. Okay, so that can be done, that can be manipulated, or you can say regulated, genetic manipulation. Okay, now let us see about the mechanism of nitrogen fixation, how nitrogen fixation is carried out. Well, if we see the mechanism of nitrogen fixation, we have to go through this part, and let us see. Uh, Very easily, it has been mentioned here. What happened during nitrogen fixation? Let us see. It is saying that inside the root nodules of leguminous plants, the bacteria will proliferate to grow. So, inside the root nodules of leguminous plants, the bacteria, that is rhizobium, bradyrhizobium, all that are symbiotic nitrogen fixers, they will proliferate, they will grow. These bacteria exist in a form that has no cell wall. So, these bacteria that are living inside the nodules of leguminous plant, they don't have any cell wall. The bacteria of the nodules are capable of fixing nitrogen by means of a specific enzyme known as nitrogenase. So, nitrogenase is the specific enzyme which is needed by the bacterial cells or the symbiotic nitrogen fixers to fix that nitrogen. So this is an important enzyme which is needed during your nitrogen fixation process. So you have seen here what I have said. So normally within the nodules of leguminous plants, or you can say legumes, the bacteria grow. Now those bacteria which are living inside the nodules of the leguminous plant, those bacteria which are living inside the nodules of leguminous plant, they don't have cell wall. Okay, inside, not outside. Outside they have cell wall, but those special nitrogen fixing bacteria which are found inside the nodules of leguminous plant, they don't have cell wall. The bacteria of these nodules are capable of fixing nitrogen by means of specific enzymes, namely nitrogenase. Okay. Now let us see how nitrogenase, which are found inside the bacteria, how the nitrogenase carry out the nitrogen fixation. Well, it is a complex enzyme. That means it has two components. Many components are there. Some components are there which carries out the nitrogen fixation. Let us see very carefully. It is a very easy concept. If you see, well, you know about reduction and oxidation. Before going into this term, before going into this nitrogenous function, we have to go through this term, reduction, oxidation. Well, you know these two terms, you have learned in your chemistry classes. From class 9 you have studied reduction and oxidation. Oxidation means addition of oxygen or removal of hydrogen or removal of electron, whereas reduction means the opposite of oxidation, addition of hydrogen, addition of electron or removal of oxygen. So here in nitrogenous they will carry out this reduction and oxidation process. How? Let us see. So nitrogenase is a complex enzyme. It has two oxygen sensitive components. So again I repeat, nitrogenase is a complex enzyme. It has two components which are oxygen sensitive. Name of that components are component 1 and component 2, right? Component 1 and component 2. What contains in component 1? Component 1 has two alpha protein subunit. Component 1 has two alpha protein subunit and two beta protein subunit. It has 24 molecules of iron. It has 22 molecules of molybdenum, which are represented by MO, and an iron molybdenum cofactor. Well, cofactor helps in catalyzing the reaction. Okay, together it is called as FeMoCO. Again, I am repeating, nitrogenase is a complex enzyme. It has two components which are oxygen sensitive, component 1 and component 2. Well, we have to first study about component 1. Component 1 has two alpha protein subunit and has two beta protein subunit. It also consists of 24 molecules of iron, represented by here as Fe, two molecules of molybdenum, represented by MO here, and an iron molybdenum cofactor, which is represented by CO. Together, it is known as FeMoCO. Let us see the component 2 now. Component 2 have two alpha protein subunit, which is fully different from the component 1 alpha protein subunit. Again I repeat, component 2 have two alpha protein subunit, which is completely different from the alpha subunit found in the component 1, and it have a large number of iron molecules. Okay. Okay, I think you have understood the components found in the nitrogenous. Now what it does, component 1 of the nitrogenase catalyzes the actual conversion of nitrogen into ammonia. The component 1 found within the nitrogenous enzyme complex, this component 1 carries out the main conversion of nitrogen into ammonia, while the component 2 donates the electron to component 1. Again I repeat, component 1 of nitrogenase catalyze the actual conversion of nitrogen into ammonia, whereas component 2 donates the electron to the component 1. Okay, so this is the structure of your nitrogen fixation. This is the nitrogenous complex, as you can see here. Well, you can see here the component 1 and 2 has been mentioned here. Okay, so component 1, what it does, component 1 is mainly they are catalyzing the formation of nitrogen into ammonia. So they are main responsible for conversion of nitrogen into ammonia, while component 2 is actually donating the electrons to the component 1. It is donating the electrons to the component 1 to carry out the function. Now when component 2 is donating the electrons to the component 1 becomes reduced, and with the help of that electrons, component 1 is converting that nitrogen into ammonia. Okay, simple thing. Just go through this diagram. More to go now, more information will be provided now in this nitrogen fixation. Now we will come to a new term called leg hemoglobin, which is actually an important component found in the nodules of leguminous plant. Well, when I have said hemoglobin, that means it will have a red in color. So again I'm repeating, so for that to easy to understand, component 1 is carrying out the main process. It is carrying out the main process, that is it is converting the nitrogen into ammonia, whereas component 2 is providing the electrons. It is providing that electrons to component 1, so that component 1 become reduced now, because reduction means addition of electron or addition of hydrogen or removal of oxygen. So component 2 is providing electrons to component 1, and using that electrons, component 1 is converting that nitrogen into ammonia, and this cycle is goes on and on and on. Okay, now I will go through this important part called leg hemoglobin. Well, leg hemoglobin, what does it do and where it is found? Leg hemoglobin is a protein which can be compared to the hemoglobin found in animals, and this leg hemoglobin is found in the nodules of your leguminous plants. Leg hemoglobin contain iron, and that's why it is red in color. It is a oxygen binding protein because hemoglobin generally bind with oxygen. The heme group or the heme part of the leg hemoglobin is synthesized by the bacterium, while the protein part, which is the globin portion, is produced by the host plant. So it's a peculiar thing. In leg hemoglobin, the heme part, the heme part is synthesized by the bacterium, which are symbiotically associated with the plant, whereas the globin part, which is the protein part, is synthesized by the host plant, that is the leguminous plant, and is red in color. Now why this leg hemoglobin is produced? Leg refers to the, here the leg is considered as the root. Okay, that's why the name comes leg hemoglobin. Leg hemoglobin is absolutely necessary for nitrogen fixation. Leg hemoglobin is specially needed for nitrogen fixation. During the process of nitrogen fixation, those nodules, those nodules of leguminous plant, those who don't have the leg hemoglobin, they are not capable of fixing nitrogen. Why? That has been given in the next paragraph. Why the nodules, those who don't have this leg hemoglobin, uh, cannot fix the nitrogen? Why? That has been mentioned in the your in the next paragraph. Let us see. It is mentioned that It is mentioned that it is the leg hemoglobin which facilitates the appropriate transfer of oxygen. Normally, the transfer of oxygen to make the components oxidize and reduce the nitrogenous components, right? So, normally, leg hemoglobin facilitates the appropriate transfer of oxygen to the bacteria for respiration to produce ATP. Well, the bacteria has to need oxygen for the generation of ATP. Well, respiration is specially needed by all of the living organism to produce the ATP, which are the energy currency of the cell. And with the help of ATP, that will be utilized for various metabolic functions. Right? Understood? So, leg hemoglobin facilitates the transfer of oxygen to the symbiotic bacteria so that they can carry out their respiration and produce energy and produce ATP. And energy in the form of ATP is absolutely required for nitrogen fixation. This is the first function of leg hemoglobin. So why leg hemoglobin is produced? Leg hemoglobin is produced so that they can transfer the oxygen and provide that oxygen to the bacteria which are associated with the nodules of leguminous plants, so that the bacteria can use that oxygen for respiration purpose, and they will produce energy from them and they will survive. Another important function of leg hemoglobin is that it prevents the damaging effect of direct exposure of oxygen on nitrogenous. Well, nitrogenous is very sensitive to oxygen, as it has been written in the first line that nitrogenase is a complex enzyme. It contains two oxygen sensitive components. Why it is written oxygen sensitive? Because nitrogenase nitrogenase can get damage on direct exposure of oxygen. So to prevent that damage, leg hemoglobin leg hemoglobin easily what it does, it transfers that oxygen. So nitrogenase does not get that oxygen, but that oxygen will be provided to the bacteria, so that bacteria can able to utilize that oxygen for generation of ATP as a result of respiration. Okay. So, leg hemoglobin is a special component which is prepared from plants as well as from the bacterial cells. The bacterial cells produce the heme part, whereas the globin part, the protein part, is produced by the leguminous plant and forms leg hemoglobin. Well, those plants, those nodules of the leguminous plant, those who don't produce this leg hemoglobin, they are not capable of fixing your nitrogen. And it has been said that leg hemoglobin facilitates the transfer of oxygen so to the bacteria, so that bacteria can able to do the respiration and produce ATP. And on the other hand, leg hemoglobin also preventing that oxygen to get transferred to the nitrogenous complex, because nitrogenous complex are highly sensitive to oxygen. Okay. Now in this figure 52.2, if we see, in this figure 52.2, if I wholly I show you. So in this figure, the fixation of nitrogen by symbiotic bacteria is given here. Okay. Now as the oxyhemoglobin supplies that oxygen, you can see here the oxygen is carried by the leg hemoglobin and becomes oxy leg hemoglobin. So when the oxy leg hemoglobin supplied that oxygen, the bacterial respiration occurs. It produces ATP from the ADP. Okay. Now the ATP that is generated during bacterial respiration, The ATP that is generated during bacterial respiration, that ATP will be used for fixing the nitrogen to produce ammonia. The ATP which is generated during bacterial respiration, that ATP will be used for fixing the nitrogen to produce ammonia. Now the whole complete reactions, if I if I if I show it, if I try to show it to you here, it will be like this. It has been given here, no worry, but the whole reaction will be uh, in this way. If I show you uh, nitrogen, okay, little bit, okay, nitrogen plus 8 protons plus 8 electrons plus 16 ADP molecules. Okay, 16 ADP molecules will give you will give you 2 molecules of ammonia. That means it has been fixed. The atmospheric nitrogen has been fixed here, plus H2, that is hydrogen. Now this gas will be liberated out from the atmosphere into the atmosphere, plus 16. Okay, sorry, I have written a little bit wrong. It will be ATP. It will be ATP. Sorry, that ATP will be utilized, right? 16 molecules of ADP plus inorganic phosphate, 16 molecules of inorganic phosphate, because 16 ATP has been utilized to form ADP, so 16 inorganic phosphates are being formed. So you can see that nitrogenase, how it carries out the whole reaction. So you can see here that one molecule of nitrogen, 8 protons, electrons, and 16 molecules of ATP. This ATP is produced during your respiration process. respiration Respiration process, sorry for that, respiration process, to be used and produce these 2 molecules of ammonia, hydrogen gas which is liberated out into the atmosphere, inorganic phosphate, and ADP molecules. So in this way your nitrogen is getting to be fixed. So the whole fixation of nitrogen by symbiotic bacteria as given here, when the oxyhemoglobin supplies that oxygen, um, sorry, not oxyhemoglobin, oxyleg hemoglobin is supplying that oxygen, bacterial respiration occurs to form this ATP. Okay, to form this ATP, and as the ATP is generated, um, that ATP will be used for fixing the nitrogen into ammonia. Okay, so this is the main reaction that you have seen uh, during this process, and the whole nitrogen fixation has been given here as done by the symbiotic bacteria. So you have to just remember this diagram. This is the simplified diagram. I hope you have understood this part what I have tried to say. Okay, more to go, because we have done the symbiotic nitrogen fixation, but manipulation can also be done. That has to be done today, so there will be a lengthy lecture little bit. So let's do the next part. Now there's another enzyme which is found during the course of nitrogen fixation, and that is your hydrogenase. So during the course of nitrogen fixation by nitrogenase, an undesirable reaction also occurs. So when your nitrogen fixation occurs, and that nitrogen fixation catalyzed by the nitrogenase enzyme, some undesirable reaction also going to be occurred, and that is your reduction of hydrogen into a proton into hydrogen gas. Well, we have seen to now, right? We have seen here that the protons are converted into hydrogen gas, we have to utilize this hydrogen gas. We have to utilize these protons. Otherwise, it is a bit of energy loss. It is a bit of loss. We have fixed the nitrogen, that's good, but we are also losing the protons here to form hydrogen gas that goes out to the atmosphere. So, so this undesirable reaction is reduction of the H plus ion to hydrogen gas. So simply the protons are going to be reduced to form hydrogen gas during your nitrogen fixation process. Now for the production of hydrogen, ATP is used, rather wasted. Okay, so the ATP is also utilized for the production of hydrogen gas, right? So we have to prevent this. We have to prevent this because ATP is getting utilized, because we have to stop this reaction, then only we can conserve our ATP. Okay, consequently the efficiency of nitrogen fixation is drastically lowered. Obviously, if more ATP is invested on this reaction, if more ATP is invested on conversion of protons into hydrogen, then automatically your nitrogen fixation will be drastically lowered. Okay, you have given money, for example, you have given money to spend the things, so you will try to utilize that money, right, rather than spending in a lustful manner, right? So for that reason here also we are doing the nitrogen fixation, that's good. We are doing the nitrogen fixation as given in this reaction. We are doing the nitrogen fixation, that's good. The nitrogen is getting converted into ammonia, but at the same time if you see, the hydrogen gas is being produced from the protons, and for conversion of these also we also need ATP, and for conversion of nitrogen into ammonia we also need ATP. Okay, and this is the main reaction that we require. We don't require this one. This is the unnecessary reaction. So we have to stop this, right? We have to stop this. So for the production of hydrogen we using ATP, okay, rather than wasted. So thus consequently your efficiency of nitrogen fixation is drastically lowered. Okay, now it is possible theoretically that how to reduce the energy wastage by recycling the hydrogen to form proton. So it is saying that how theoretically, practically it is possible, but we have to manipulate the genes here. So how you can recycle that process? That means we will convert the hydrogen into protons. How? To liberate energy. It is saying that there are some strains of Bradyrhizobium japonicum. There are some strains or families of Bradyrhizobium japonicum in soybean plants were found to use hydrogen as electric energy source. So some scientists have observed that there are some species, there are some strains of Bradyrhizobium japonicum that associate with the soybean plants as legumes, and these Bradyrhizobium japonicum were found to use hydrogen as the energy source. They don't use ATP, they use hydrogen as an energy source. These strains were found to possess an enzyme, namely hydrogenase. So they have an enzyme called hydrogenase which catalyze this process. They use hydrogen as energy source, and they use that hydrogen as energy source, and they use that energy for fixation of atmospheric nitrogen. Recycling of hydrogen gas that is formed as byproduct in nitrogen fixation is also shown in the figure. So if you see the figure 52.3, you will see that how the hydrogenase enzyme is using that hydrogen is using that hydrogen as energy source by recycling the hydrogen to form H plus ion. Let us see the figure. Okay, this is the figure. So you can see here, this is the nitrogenous complex, the nitrogenous complex that we have studied little bit in detail in figure 52.2, where it consists of complex 1 and complex 2, okay, whatsoever, but the main end products are ammonia and hydrogen. This hydrogen, this hydrogen is again recycled by this hydrogenase enzyme, which is found in some strains of Bradyrhizobium japonicum within the soybean plant legumes. So these hydrogenase can recycle and use this hydrogen as energy source. So you can see here they are using that hydrogen to produce protons, and they as electron carriers they are using them into the nitrogenous complex, where from component 2 it donates that electron to component 1, and component 1 will utilize that electron for conversion of nitrogen into ammonia. And this is actually the respiration of the bacterial cells, not respiration, sorry for that, this is the respiration if I do the aroma like this, but this is the utilization of ATP. This is the utilization of well, wait for a minute. Okay, now okay. So this is the utilization of ATP to use ADP, because ATP is utilized for conversion of nitrogen into ammonia. At the same time, the protons are utilized to form hydrogen by using the same ATP. So ATP is getting, I don't say wasted, but it is utilized. But we have to recycle this, because otherwise the efficiency of nitrogen fixation will be drastically lowered. To prevent this, it has been observed that some strains of Bradyrhizobium japonicum, they have this hydrogenase enzyme. What they do is they use this hydrogen, as energy source. Okay, and they use this hydrogen as energy source, and they utilize that hydrogen to produce protons again, but that means they're recycling, and these protons with the help of electron carriers are transferred to the nitrogenous complex, where they will use the same process. They will transfer the electrons from component 2, it will donate that electron to component 1, and component 1 will become reduced, and during in reduction form, this component 1 will convert that nitrogen into ammonia. So in the same way we are utilizing the energy, but it is found in certain strains, like in Bradyrhizobium it is found, right? Okay. Now it is saying that it is advantageous for nitrogen fixation if the symbiotic bacteria possess the enzyme hydrogenase. Now it is saying that if the symbiotic bacteria have hydrogenase enzyme also with nitrogenase, it will be beneficial for them. So those organisms, now think as genetic engineering, as I have mentioned with you in the previous lecture of during your biotechnology and human welfare lecture, that if somehow, if we can introduce the gene, if somehow if we can introduce the gene that will code for this hydrogenase enzyme, where we will introduce them to the nitrogen fixing bacteria, which only produce nitrogenase. Then what will happen? It will produce nitrogenase as well as the hydrogenase. So more efficiency will be taken place for nitrogen fixation. This is the genetic manipulation. We are regulating, we are modifying the genetic makeup of that microbes here by introducing certain genes or by blocking certain genes. Here we are introducing certain genes. So it has been mentioned clearly that it is advantageous for nitrogen fixation if the symbiotic bacteria possess the enzyme hydrogenase. So if the bacteria which is symbiotically associated with the plant, if they have the enzyme hydrogenase, there will be more efficiency in the process of nitrogen fixation. However, naturally occurring strains of Rhizobium and Bradyrhizobium do not normally have this hydrogenous enzyme. The gene responsible for the hydrogenous enzyme is not present in normal Rhizobium or Bradyrhizobium. It has been observed in some strains of Bradyrhizobium japonicum. Some few varieties of Bradyrhizobium japonicum have this hydrogenous enzyme capability. Okay, it have this hydrogenous enzyme capability, right? Okay, so what to do to increase the efficiency? Genetic engineering can be done. We will isolate the genes responsible for the responsible for that hydrogenous enzyme capability, and we will introduce that gene into the into a particular region of the genome of such nitrogen fixing bacteria. The gene will get expressed in that bacteria, and the bacteria will able to synthesize the hydrogenase along with the nitrogenase, and thus we can increase the efficiency of the nitrogen fixation. Okay, so this was all about the nitrogen fixation. Okay, now because as it is given related with the approach related with the biotechnological and human welfare approach, so we need to do this part also, genetic manipulations for the nitrogen fixation, how we can increase the nitrogen fixation process efficiently. Okay, so let us discuss about the genetic manipulations for nitrogen fixation. It has been mentioned that nitrogen fixing bacteria that are closely associated with the world's food supply are among the favored organisms for genetic manipulations, because most of the foods, protein rich foods, are dependent on nitrogen fixation, and for that we need to fix the nitrogen, we have to improve the strains of nitrogen fixing bacteria. So biotechnologists have considered the following possibilities for effective utilization of diazotropes. So we can enhance the more production of diazotropes, the symbiotic nitrogen fixers and also asymbiotic nitrogen fixers as natural biological fertilizers. So this is the main aim for our improving the crop cultivation, improving the those crops which are which will be supplying as a protein source, which are the main of the world's food supply. Okay, and not by using the chemical nitrogen fertilizers, by just using the biological nitrogen fixers, we will supply that nitrogen in our soil, will provide them into the plants. Okay, and how it can be done through the help of biotechnology. Well, I have said you have also know during your class 12 syllabus, you have also studied a little bit about biotechnology, and I have also said that how genes can be inserted into the certain living organisms, like I have said in case of plants also. So here what we can do, the first way that we can do is we can alter our gene, we can modify the gene in Rhizobium species to improve nitrogen fixing efficiency and bacteria host plant interactions. So by modifying the genes, by introducing certain genes into the rhizobium, like for example we have introduced the gene which is responsible for the encoding of your hydrogenase enzyme, and hydrogenase enzyme is specially needed so that we can recycle the hydrogen into protons to be used for the nitrogen fixation, so that you can prevent the wastage of energy. Okay, so this type of gene alterations can be done, or you can modify the genes so that the that particular species can more interact with more species of the plants, even non-leguminous plants it can interact. This type of genetic alterations can be done. Okay, to improve the nitrogen fixing efficiency and bacteria host plant interactions. Second is genetic engineering of Rhizobium species so that it can form a symbiotic relationship with the non-leguminous plants. Just I have said just now that we can modifying the gene present in the rhizobium. Why we are talking about rhizobium? Because there are many variety strains of rhizobium that can be used to interact with the plants. Like we have seen that rhizobium interact with the wait rhizobium species interact with the pea, broad bean, alpha soybean, depending on what type of rhizobium species we are using. Rhizobium loti is associated with the lotus. So selected plants are involved, selected host leguminous plants are there which interacts with the rhizobium. But through genetic engineering technique, we can modify the rhizobium species by introducing certain genes um, that will encode some enzymes so that rhizobium can interact with the non-leguminous plant as well, right? So that is the thing that they want to say, that genetic engineering of rhizobium species can be done so that it can form a symbiotic relationship with the non-leguminous plant like wheat, rice, corn. These are non-leguminous plant, and they also need nitrogen, by the way. But to get the nitrogen, naturally there is no such nitrogen fixing bacteria which associate with the wheat, corn, or rice. So to get the nitrogen biologically, we can modify the rhizobium species so that they can associate with this type of non-leguminous plants and can fix the atmospheric nitrogen, right? Next is your transfer of genes for nitrogen fixation from rhizobium species to other bacteria as well, like Agrobacterium tumefaciens. Well, Agrobacterium tumefaciens, I have little bit discussed with you that this bacterium, they infect the plants and form crown gall disease. Okay, and now and these agrobacterium tumefacients are best for genetic engineering because they have a plasmid. Within this agrobacterium tumefacients, they have a plasmid. Plasmids are extracellular circular double stranded DNA which are capable of independent replication. These plasmids can be used as a vector, as a carrier molecule for inserting our gene of interest into a host plant. So we can transfer the genes for nitrogen fixation from the rhizobium species to the other bacteria. The so developed Agrobacterium tumefaciens can infect several important crops like tomato, tobacco, petunia, and fix the nitrogen in the host plants. So what we are doing, the genes which are found in the rhizobium, their genes are isolated and they are allowed to be introduced into the agrobacterium cells. Obviously, we will remove the pathogenic part of the agrobacterium that cause the infection to the plants, and we introduce our gene of interest used for the nitrogen fixation into that agrobacterium. Now that modified agrobacterium cells are allowed to infect the plants. The agrobacterium normally, the way that does the infection, it will carry out the infection process, and at the same time it transfer that gene into the or group of genes into the host plants where the genes will get expressed and will carry out the fixation of nitrogen. So very wellly, it is theoretically it is very good to be mentioned, but practically it is bit of difficult task if I say, because uh, to to manipulate the genes to express the for expression of that genes into the host plant is because it is a foreign gene now, by the way, and it's a difficult task. Okay, so practically it is a little bit difficult. It's bit challenging, better to say, not difficult, better to say challenging, but it can be done and can be in used to infect several non-leguminous crops like tobacco, tomato, petunia, and fix the nitrogen. So in three ways we can do: either we can alter the genes to the rhizobium, and then we use this rhizobium to interact with the leguminous plant, okay, to increase your nitrogen fixation. Like for instance, if we can allow the genes for hydrogenous production into the rhizobium species, because most of the species, no, most of the nitrogen fixing bacteria, they don't have hydrogenous. Okay, so, if we can able to use this hydrogenous gene, insert that hydrogenous forming gene into the rhizobium species, and if that gene get expressed, then more nitrogen fixing capability will be provided by that particular modified strains of rhizobium. Second, it has been mentioned that genetic engineering of rhizobium species, so that it can form a symbiotic relationship with the non-leguminous plants. So we can modify the gene, we can modify the genome of the rhizobium, so that the rhizobium can able to able to interact with the non-leguminous plants as well, like wheat, corn, rice, etc. And the third is bit interesting. It is in saying that we can transfer the genes for nitrogen fixation from rhizobium species to other bacteria like Agrobacterium tumefaciens. So normally what happened, I think I have mentioned with you in your when I was talking about applications of biotechnology in agriculture. Uh, well, Agrobacterium tumefaciens is a gram-negative bacterium found in the soil, and this Agrobacterium tumefaciens can infect the host plants. It can infect certain dicotyledonous plants. Now, what we can do is, if I okay, so within this agrobacterium cells, it there contains a plasmid. Okay, that plasmid known as the Ti plasmid or tumor inducing plasmid. Now, this Ti plasmid contains some pathogenic genes. This Ti plasmid contains some pathogenic genes which are normally used to be transferred to the host plants to get infected with crown gall disease. Now, what we can do, we will isolate that Ti plasmid. We will isolate that Ti plasmid. We will then, what we can do, we can eliminate that Ti plasmid. Okay, wait for a minute. We can isolate that Ti plasmid, and then we can eliminate that Ti plasmid, that pathogenic part which is causing the infection, and in that place we can introduce our gene of interest. We can introduce our genes needed for nitrogen fixation into it, and then we again, what we do, we again then implant them into the agrobacterium cells, and that agrobacterium cells, wait for a minute, that agrobacterium cells will now contain that modified plasmid, will now contain that modified plasmid. Okay, now agrobacterium cells is allowed to infect to the host plant. It is now allowed to infect to the host plant. Okay, then what happens? These plasmids will get inserted to the genome of the host plant, but we have removed the pathogenic part, and it contain our genes for nitrogen fixation capability, so that the so developed agrobacterium tumefaciens can infect several important crops and deliver these nitrogen fixing genes into the plant's cells. Okay, thus the plant cell will itself produce that nitrogenous enzyme, will produce that hydrogenous enzyme, and carry out the nitrogen fixing process in order to get such type of plants. So I hope I have tried to understand that part. Okay, okay, okay. So Some other considerations are also being used nowadays. Some other considerations are also being nowadays, like insertion of nitrogen fixing genes into plants directly, or even animals and including humans. We can also introduce these nitrogen fixing genes directly into the plants by direct gene transfer, like micro injection is there, biologistic method is there, that is a gene gun method, which I have discussed a little bit with you. Okay, so that type of process can also be done directly. We can insert our gene, the nitrogen fixing genes into the plants directly, or even animals including humans. This approach, which enables the plants, animals, humans to directly trap and utilize that nitrogen from atmosphere, is rather theoretical. Now, theoretically, it is possible to say that if we can insert the genes into the plant, the gene responsible for nitrogen fixation, if it is inserted into a plant, and if the plants express that nitrogen fixing genes, then what happened? It will enable the plants to directly trap that atmospheric nitrogen and utilize the nitrogen for the production of amino acids and proteins. Okay, and also the nucleic acids. So, but it is theoretically possible. Practically, it is challenging. Life would be certainly different if the production of nitrogen fixing genes in man can dispense the habit of eating daily. That means if this becomes successful, well, now research is going on to deliver the genes into the plants, and the plants are expressing that genes, right? If that same nitrogen fixing genes are tried to be incorporated to the human beings, and if the human beings capable of expressing that nitrogen fixing genes, that means they will take that atmospheric nitrogen from the air and fix within their body to make ammonia and ammonia like compounds, and they will then use them for the production of nucleic acids, for the production of proteins. Then what will happen? Life will be different, because we don't need have to eat, because we get proteins from them. In a similar way, if we can get energy, okay, so there will be dispense of habit of eating daily, so our daily food requirement will be much lower, because we can synthesize our own proteins and amino acids by fixing the atmospheric nitrogen within our body, by inserting the genes into our body. Okay, the nitrogen fixing genes, but that will be a ethical issue. Firstly, the first most important that will be occurring, this ethical issue will be occurring. Even the gene therapy which is being used nowadays to treat certain genetic disorders is now becoming a ethical issue. Okay, so why, if we introduce this nitrogen fixing genes into human, then ethical issue will occur. Different types of communities will raise voice, and there will be lots of riots, revolts will be going around. Okay, so this is theoretically possible, but nowadays it is trying to insert the genes into plants to genetically modify the plants. Even there also there is ethical issue. Despite several possibilities and optimistic predictions, the success in genetic engineering in relation to nitrogen fixation is very limited. There is very limited way that how nitrogen fixation is done through genetic engineering and allow the improvement of the strains. Okay, that is very rarely mentioned here, but how it can be done, it has been said theoretically, and practically it is being nowadays done in plants, not in animals and humans, right? So we will go into this in detail. Okay, so that's all for today's lecture. We will go this detail later on. So actually this has been up to this much I have mentioned with you with this lecture. Okay, you can go for the lectures. I have given you the PDF of these notes, wholly fully I have given. Okay, So just go through these notes, to up to what I have done today. And this part, I don't think it will be given in your syllabus. It is a exceptional part. The nitrogen gene cluster, these all parts. Well, if it is given in your syllabus, then I will try to discuss this with in the next lecture. Okay. So that's all for today's lecture. If you have some doubts regarding this part that I have mentioned with you all today, you can ask me in the WhatsApp group. Okay. So thank you.